Welcome all. It's gone 8 o'clock where I am, maybe 6 where you are, or any time depending on where you are. Mark Ferguson, Next Gen Agri. Thanks very much for joining us tonight. We're joined by Sarah Preston from SwabTec. Welcome, Sarah.
Hi everyone, and thanks Mark for having me.
Well, good. It's really Sophie who's had us both, really, but we're here to have a chat. So I guess just rules of engagement. Ask your questions on. There's live Q&A, so there's the little question mark symbol down the bottom. There's also chat. You can chat away as well. I'm not 100% sure why we got both of them, but anyway, ask your questions in that, and I'll once Sarah's up and running, I'll try and relay them to Sarah as we go. Exciting bit of technology that we're chatting about tonight. Obviously, depending on where you are in the world, there's significant issues with drench resistance and managing worms, and we've had some wet seasons in some parts of the world, and particularly where I am, and having some really serious worm burdens, which brings a lot of pressure on the system. And so this technology is obviously all about helping us breed sheep to be more resistant. Sarah, do you might want to just give us a quick background of your background, and then we'll hook into your presentation.
Yeah, no worries. So I am based or joining you from Ballarat today in Victoria in Australia. So I work out of Federation Uni Emma, I've been an animal health researcher for 13 years. I also teach students in a Bachelor of Veterinary and Wildlife science course as well. And I guess, I grew up in a sheep and cattle and cropping farm in New South Wales, Australia. And I guess that's where my passion for becoming an animal health scientist came from. Just sort of last year formed the company SwabTec so that we could look to sort of commercialise some of our research that we'd been working on that will allow farmers to breed more worm-resistant sheep so that you don't have to be so dependent on drenches.
Cool, thanks, Sarah. And feel free to introduce yourselves on chat if you want to have it. There's a few people doing that. Thanks, Sophie. We'll ask Sarah to bring up your presentation and we'll wander through that. Just put your questions in the chat as we go, and we'll endeavour to answer them. Or I won't, but Sarah will.
Hopefully everyone can see that.
Yeah.
Nice and clear. I'm just working off one screen today, so I can't see if there's any questions as I talk through the slides, but of course happy to answer them either after the presentation or during. I'll let Mark sort of interrupt me as I go through, but I am the co-founder of SwabTec, and what we're developing and validating in Australia is a simple saliva test to allow farmers to start to breed for worm-resistant sheep. So I guess I thought I'd start with a little bit about where the company came from, and I've already introduced myself, essentially we're based in Victoria, Australia, and we just formed last year, again, just to try and commercialise some of the research out of the university. So it is a family-owned business, so I co-founded it with my partner who has a background in business management and marketing and product launching. To largely rely on casual workforce at the moment. So I've got Elizabeth, Tanya and Rebecca. These are all my PhD students that are working on other important worm projects, and they can jump into the lab and help with bits and pieces when I need extra support or come on to farms with me. So I'm lucky to have such a good casual workforce to support this company. We also have some really key advisors that help us navigate sort of the commercialisation space. So that's Professor David Pietrafita, who's been a parasitologist for many, many years. We have George Wilkes as well, who used to work for Zoetis, and Vern Bowles, who's also a great parasitologist and immunologist. So that's SwabTec, and that's who we are. I thought, I'd just start with this sort of slide, which is probably no surprise to anyone on this call, just about how much worm infections do cost the sheep industry. So the latest MLA report has costed, for at least for Australia, it's $600 million a year that we're spending on largely drenches to control worms. That number also looks into production loss as well. And as Mark brought up, there is a lot of drench resistance around. Some places in Australia, particularly New England area, they don't have any products that work anymore, and they have had to switch to cattle. Those are sort of the stories that are coming out. And unfortunately, we still don't have a multi-strain vaccine. We do have a vaccine in Australia called Barbavax that will protect against Barber's pole. But we're sort of yet to have that multi-strain vaccine for worms. So I guess one way you can try and help alleviate the worm burden and the worm problem is to start to breed for worm-resistant sheep. And those studs that have been doing this for many, many years have found that their sheep are more profitable, that they're not having to intervene with drenches, and they're not seeing production loss. And there is an increased demand from commercial sheep producers to look for rams that have good worm resistance as well. So that's been a trend that AWI have reported on. In Australia, unfortunately, we only have one marker that allows producers to start to breed worm-resistant sheep, and that's the worm egg count. We have a threshold of 300 eggs per gram in Australia. That means you need to meet that before you can start to measure individual sheep. And so with some of the regions, depending on their worm species that they have, reaching an egg threshold of 300 eggs per gram is quite high, and so they do see production loss and have seen death in trying to implement this. And some of our studs that have been breeding worm-resistant sheep for many, many years and have highly worm-resistant sheep, they essentially have to make their sheep wormy to get an infection so that they can measure, so really pushing their sheep, and then maybe in a month's time turn around and try and sell their rams, so they have to then refeed them. So it's pretty counterproductive for those studs. So another way you can think about breeding worm-resistant sheep is actually measuring their immune response to worms and actually, measuring immunity. And this is not a new thing. I think New Zealand's been really lucky to have the CALA tests that they have, so that allows producers to look for animals that are producing high levels of antibodies. The UK also has a blood test now too, or an antibody test. Theirs is a blood test and I think that's particularly in the Lynn sheep, if I'm pronouncing that breed right. And these tests both sort of target the L3 stage of the worm, so that's the stage on pasture, and as they get ingested, that's sort of the antibody response that these tests are targeting. I thought before moving into the tests that we've developed at SwabTec, I'll just take you through a bit about the immune response to worms. So this is sort of animation, which is obviously not to scale, but obviously as the sheep start to ingest the pasture, that's when they get infected with this L3 stage. And roughly it takes 24 to 28 hours to reach the area in which they like to inhabit, which is the abomasum for some of the worm species, and then the small intestines for other worm species. And as they are migrating to those areas where they start to develop into adults and through a few different juvenile stages before they get to adults and start laying eggs, they're actually secreting a lot of molecules. And these molecules, in science, we call them the excretory-secretory molecules. And a lot of these molecules that are released actually being shown to switch off the immune response or dampen down the immune response. And that allows the worms to sort of go undetected in the sheep and allows them to establish an infection and then breed and create the eggs that then go into pasture. So as the sheep is trying to defend against the worms are trying to defend against the host immune response as well. And as they start to defend against or secrete these molecules to defend and shut down the immune response, it allows them to grow from the L3s to the L4s and then the adult stage. And that roughly takes around 21 to 28 days before you start to see egg production from that stage of infection. But there is a lot of environmental impacts about the length of time. And some of the worm species can go into a dormant phase inside the host as well. And then just sort of the last slide about the immune response. I just wanted to highlight that although we talk about measuring an antibody response to worms, it also means that you're breeding sheep that have a really important cellular attack as well. So you're not just identifying sheep that have only depend on an antibody response to worms. So when we talk about immunity to worms, sheep that are known to be immune, they've got a really highly orchestrated, integrated attack against the worms, and this involves both the cellular response, so they recruit certain cells to do damage to the worm, but also an antibody response that work together with the cells as well. And in many, many papers that have been published, and this is not everything that an immune sheep will have, but the responses that keep coming up quite regularly in immune sheep across many different breeds is that they produce a lot more mucus, and so that's the yellow part of this diagram. So should highlight that this is my attempt to draw the sheep's gut, and the cells are coming to attack this worm here. They'll also have higher antibody production, in particular IgA. So that's a really important antibody response that's produced in the gut. And there's been many studies showing that sheep with high levels of IgA in the gut will have reduced number of adult worms. That's been shown in many breeds as well. And the immune sheep will also have many or higher levels of immune cells, and particularly this type of immune cell, which is a eosinophil, which actually binds to the worm and degranulates or blows up essentially to do damage. So I just wanted to highlight that when we're trying to identify immune sheep, they not just don't have this particular antibody response, they've got a whole range of different responses that they rely on. And then that ultimately results in a decrease in the number of worms, particularly in infections with barber's pole and also the scaleworm species. And with the brown stomach worm, that usually the response to that is more around reducing the size of the worms and then therefore egg production. So based on that, we've developed an antibody test at SwabTec. So we've been able to show with the help of AgBrew that it does have a good heritability. That's by testing over 2,000 animals so far. And what we've targeted is one of these molecules that are secreted by the worm. So these molecules that actually shut down the immune response. And so we're trying to identify sheep that produce high antibodies that are able to bind to these molecules that shut down the immune response to neutralise that attack and essentially prevent the worm from being able to switch off the immune response or dampen down the immune response and allow the infection to continue. The other reason why we chose this type of molecule as well is that we can make it synthetically really easy. It's quite stable too, and there has been previous work by other groups showing that it's a key response in worm-resistant sheep as well. We do have a provisional patent application on this technology, so I can't actually release any more like the name of the molecule. Eventually we will be able to, but that does need to be kept a secret for now. The antibody response that we are measuring is an IgA response, and that is linked to the fact that, there's been many studies showing that if you can breed sheep that have high levels of IgA in the gut, you will have sheep with lower worm burdens. And you can measure this in the saliva, just like with the Carla test. And the other thing we like about this test too is that you're measuring sort of this early stage in response. So these molecules are secreted by that early L3 stage, L4 stage. So you can start to measure this response much earlier than you can for WEC, because obviously WEC you have to wait for the adults for the L3 to get to adults and then start to produce eggs. So I just wanted to highlight that the next set of slides are going to be the results that we've seen so far with our test. But yeah, those sort of samples and results that we have so far wouldn't be possible without all the studs that have allowed us to test their sheep and collect saliva from their sheep. So we haven't been working in a research sort of stud or operation. We've been relying on commercial stud breeders to help us collect these results. So I'd just like to really thank them for that. So with our antibody test that we've called the worm antibody swab or wAbSwab, one of the first things we wanted to make sure was that we could measure this response without having to have a high worm challenge. So these graphs here just show you that in one flock that we measured, the worm egg count was only 50 eggs per gram. So that's really only one egg under a microscope. And you can see that even with that level of worm infection based on the worm egg count, you've got a good range of levels of antibodies in that flock to try and pick out which ones would be the more worm resistant. And even if you have a higher challenge, such as 500, you get a similar range and similar variation as well. So we know that you don't need to have a high WEC to start to measure. The other thing we've shown with this test is that it can be measured early and also not affected by drenching as well. So obviously with worm egg counts, you want the drench to work, so you want the worm egg count to decrease after a drench, but it does bring in variation in when you're trying to measure worm resistance using WEC. So in this experiment, one of the studs was able to measure the worm egg counts in their sheep when they were 5 to 11 weeks. So that was marking, weaning and post-weaning. And they also collected saliva at the same time for us. They've just used their normal worm egg count provider to get these results. And you can see when we measured our antibodies in the saliva at the same time points, we had a nice consistent increase in antibody response as the sheep aged. And we didn't have an interference with the drench, unlike the worm egg count. So that's something also that's been shown before with saliva tests as well. Now we're looking at whether our sort of worm-resistant sheep do have higher levels. So I mentioned earlier that the reason why we picked this molecule to measure from the worm to measure the response against was because it had been identified before in worm-resistant sheep as being a pretty important response to have. And that was actually, the paper for that was published in the 80s. So we're sort of building off research that's been published a long time ago. And in this...
Just quickly, Sarah, there's a question from Rodney. It's on the previous slide, so we may as well just cover it now. But with a WEC of... So Rodney's question is, with a WEC of 50, it's likely there are likely to be a lot of zero counts. Does it still work? I think.
Oh, so this one here?
Yeah, so in that left-hand version, the WEC average of 50, there'd be a lot of zeros, is what Rodney's saying, which is true. But
yeah, for a worm egg count, yep.
Yeah, but obviously the antibody there's a big range there still.
Yeah, exactly, yeah. Yeah, so we're not seeing, lots of zeros in our antibody test, even though you would see that with a WEC of 0. Yeah.
And so it's so even at a WEC of 0, they're still ingesting. They still could have adult females in the gut, or could be ingesting larvae, and that's what
is being picked up. Yeah, so this antibody response is against that early stage of the L3 worm and the L4 worm. So that's before you actually see any egg production by adults. So really measuring a response before you actually see any eggs. Yeah, which is quite important for some, like the brown stomach worm that doesn't produce many eggs anyway. And there's been some work to show that once you get to 250, 300 eggs per gram, that worm will actually stop producing eggs, but you still have worms in the gut eating from the sheep, so you're still having damage. You just can't detect that as a worm egg count. So that's, again, one of the sort of advantages of an antibody test.
Another question while we're there. Ryan Eichmann says, can you compare between farms or just within farm? Is the response L3 dose related?
So that's getting towards the next lot of, I guess, results where we're looking at how it relates to a worm egg count. I guess, perhaps.
I guess if they're ingesting heaps of L3s versus not many L3s, would that change the level of antibody?
Yeah, I guess we haven't really investigated that yet. The only way I could sort of try and answer that, because we haven't really looked at high pasture challenge, the only way we can really look at that is by looking at WEC, I guess. Yeah. And even, with counts of higher than the 500 eggs per gram, then you still get a good range and good variation of immune responses. What we're hoping to do in the next lot of trials is actually sort of measure at different time points throughout the sheep's life. I guess the only way we can really look at L3 challenge is through just the egg count results. With that sort of, you can obviously do pasture counts of larvae, but I don't think that that's super accurate at the moment.
And the other part of that question was, can you compare between farms or just within a farm? I guess the answer.
Yeah, I mean, ultimately we want to be able to have it into a strain sheep breeding value. So that way you could rank across farms, which animals perform better, but at the moment we're not quite there yet.
But you'd be confident that an animal has got a big antibody response on one farm will also, they shouldn't re-rank.
Not if it's a genetic breeding value that's been corrected for environmental effects.
Yeah.
Yeah, so just like how worm egg counts are when they're converted to breeding values.
Yeah, and we'll get to it. We've obviously done some breeding value stuff and...
Yeah.
Cool, thank you.
Thanks for those questions. So this is where we've started to look at what it looks like, what this response looks like in worm-resistant sheep. So in this graph here, we tested a stud that had been breeding worm resistance for over 20 years or so. So they have highly resistant sheep. So when we looked in their sheep that had yearly WEC ASBV of less than -60, so quite high, highly resistant, they did have high levels of this antibody response that we're measuring compared to their susceptible sheep, which you can see are not really super susceptible anyway. So that was good to see. When we're looking at worm egg counts directly with this trait, we haven't seen correlations when we measure at the same time that are significant and negative. just looking at the raw data, but what we have seen is when you measure animals, so down the bottom here we've got antibody levels, and we measured these animals at 5 to 11 weeks. So this is data from that I presented earlier from that drenching graph. So the lambs that had higher levels of antibodies at 5 to 11 weeks of age, they ended up with lower worm egg counts at this older age of 25 to 30 weeks. So that's where we see negative correlations with this trait. So there's a lag time before you see this negative response, which makes sense, I think, from an immune response that you get in getting infected. So you start to mount your immune response, and then a few weeks later, you'll end up with a decrease in that pathogen that you're trying to get rid of in terms of worms. So that's what we're seeing when we look directly with worms or with WEC. So this is correlation with WECs when we sort of measure on the same day. So if we're measuring saliva and worm egg counts the same day, we generally see this in the flocks that we've tested so far. So no significant negative correlation at all. But what we do tend to see is that the sheep that do have higher worm egg counts are pretty low at producing antibodies, whereas the ones that have higher antibodies towards the lower end of a worm egg count range. So that's sort of the pattern that we've been seeing with this response or antibody trait. So again, this is all just raw data as well, so it hasn't been corrected for anything. But what we have been able to do with the help of NextGen Agri and Sophie Barnes in particular is that we have been able to give it some of our antibody traits that we've been measuring into breeding values so that they are corrected for things like birth type and so the pedigrees in there as well. And this is just some data taken from Darren O'Brien's sheep, who I think is on the call. And I would say that this is not what we see in every flock. This is what we're hoping that we will be able to see with this trait when we do increase our numbers, and that we do see negative relationships with DAG, because we don't want to be trying to or have an immune trait that is associated with diarrhea, which some immune responses are. So we want to make sure we're not sort of breeding that type of immune response to worms with this trait. And obviously, ideally, we're looking at traits or immune responses that do correlate with increased weight as well, and not with weight loss. So really efficient immune responses that don't cause a lot of pathology, and then you get the subsequent production loss as they're trying to deal with infection. So again, we don't see this in every flock that we've looked at so far, but this was some promising results that did come from Darren's sheep. So this is the last slide, and then I can take many more questions, but essentially this is collecting the saliva, but what we've been able to show so far with our test is that we do have a relatively fast and simple non-invasive collection with the use of saliva, which again is no surprise to anyone from New Zealand that have already had a saliva test available for many, many years. We are seeing moderate heritability with this trait, which is good. What we also wanted to show was that you could measure this without a high worm infection. So you can measure antibodies and get a good range across the sheep without an egg count of 300 eggs per gram. So you shouldn't sort of see production loss if you want to measure with this trait. We have seen negative correlations with WEC, but only at certain times. So we're still trying to understand that relationship with this trait. And so we're also working with AgBoo to build up our saliva tests with our matched worm egg counts and really understand that trait from a genetic point as well. And we have seen some positive associations with health traits, but we're not seeing that across all the flocks that we've measured so far. We've also priced the test as well, so it's $8 per sheep, Australian dollars, and obviously, as I mentioned, with the help of Next Gen Agri, we can start to convert these with in-flock breeding values, so you can have a look at how it's looking in your sheep. So that's all the slides I have, Mark, so I'm happy to take further questions.
Thanks, Sarah. Is there any questions out there? I'll give you a minute to tap away.
So it'd be great to have a question from Richard. I can see Richard's on the line.
Oh, there's a couple of big ones for you.
Yes, I can see one from Fred. Just how does it correlate with growth? Yeah, so all we have at the moment with our trait against our production, other production traits that have been measured on that sheep is we have seen some positive correlations with growth. So that's what I showed with Darren O'Brien's sheep, but we haven't seen that across all sort of flocks yet. They've been working with Agri to build up a bigger database to look at those production traits genetically as well. Oh yeah, Rodney's question is there an optimal age? So again, that's what we're also looking to try and find out as well. At the moment, we're thinking any age post-weaning is probably a good time so that you avoid any potential immune responses that are being, or antibodies that are being passed on through the mum. And yeah, we're looking to go through some more trials where we're looking at those different age groups to try and work out, yeah, when would be the optimal age and if the heritability changes with the trait based on the age as well. So I guess, yeah, Rodney, we're still working out the best time to be able to measure. Obviously, yeah, I guess it's be great to be able to measure it earlier than later so that you can make your selections a bit quicker. But yeah, still not quite sure. And then I can see another question from Hamish, that the adult female worms stop laying their eggs when infections get to a certain level. Yeah, that is research that's just with the brown stomach worm. So that level has been, in published research, been shown to be around that 300 eggs per gram. So it's a species that does self-regulate its population. And so when there becomes too many adults with that species, it will stop producing eggs. So it's quite an interesting worm species, the brown stomach worm.
Thanks, Sarah. If you want to just swing over to Q&A, which is the other box, you'll get a heap of a couple of other questions in there as well. 3.
Oh yeah, so one from Richard. So the earliest stage that we've seen, that we've measured, Richard, is that, going back to the slides, it was, yeah, the 5 to 11 weeks of age, where there was low detection, but it was present, but it's, yeah, of the lowest sort of level that we can see. And then the next sort of age group that we've tested was the 25 to 31 weeks of age. So I guess we can detect it at that really young age, but the levels are quite low at that age. And Ryan's question is, any idea on the time the antibody stays elevated for? So, yeah, again, that's not something we've measured particularly for this trait, but for other antibody traits, they say about 8 weeks is sort of the stage where if you trench and treat for a worm infection, then you've got sort of an 8-week lag where you will see an antibody response. We haven't been able to, we haven't done work to actually look at that yet. But I think, your point about trying to compare with a later worm egg count is something that we're looking to do next in our next lot of trials, because that's a really good idea. Ryan? Maybe another question from Horace. On-farm, hands-on practical experience working with sheep with good selenium, cobalt, trace element levels. This supports the sheep to be strong, robust and good. Condition scores to summarise the sheep with good trace. I'll start to keep reading it in my head maybe.
I think the question bit's at the bottom.
Yeah, cool. So the question is whether the sheep immunity to worms, tolerance resistance is compromised at low levels of trace elements is low. I mean, that's a really good question and something we haven't considered at all, but definitely looking at, if there's, trace elements issues on the farm, then that definitely could have interaction with the immune response. Yeah, it's something I haven't considered, so that's a really good thought. Thanks for that. It'd be good to see how changes in larval challenge affects the response. Carla levels appear to reflect challenge. Yeah, it would be really good to see that, Richard, too. So that's something we could probably look at, in our next set of trials.
I guess ultimately we'll be looking at it as same as Carla is, as in a breeding value sense, so that essentially correcting for cohort should, in theory, correct for whatever challenge that mob has been on. And so in breeding value land, we're kind of protected against some of those subtle differences in challenge level and age and all that sort of stuff.
Yeah, definitely. Just, like how the worm egg count works across different species, and how farms are compared against farms that have Barber's pole that produces huge worm egg counts compared to others that don't have many worm egg counts. So I guess that would be the similar for this trait.
There's a question from Kim back on the Q&A around the actual collection process of saliva and where the testing's done.
So with the collection process, so there was a little video at the end slide, I don't know if you saw that. And essentially it's just 4 steps with a dental swab that you put in the side of each of the cheek, each side, and swab there for 10 seconds, or just counting to 10 is our protocol. So we've gone out to farm to help farmers do it, but also it is quite a simple thing to start to do. So we have been able to send out kits to farms that are much far away from Ballarat, and they've completed that process successfully. The testing is done in our lab in Ballarat, located at the university. And there's a question from Darren. Yeah, that's a really good question, Darren. So obviously you can measure antibodies in the saliva against other diseases too. So we have been working on that with NextGen Agri too, to have a look at what else we could be measuring as an immune trait, and start to breed that also into our sheep as more of an immunocompetent trait. So we've done some preliminary work just looking at the responses to some vaccines as well, which has been sort of exciting.
And it's probably worth just mentioning there, we've currently got a product we're calling Shield, which has, among other things, a lot of work aimed at sort of, I guess, answering lots of these questions around both the worm antibody, but also those other other disease antibodies. And so, yeah, any stud breeders that are on the line that are keen to be involved, if we are successful in getting that funded, then we'll be looking for collaborating properties on both sides of the ditch. So, get in touch with anyone at Next Gen Agri or with Sarah if that's of interest. But we will be, I guess all going well, we'll be doing a fair swag of work, really trying to bulk up this area of knowledge around sheep breeding. A couple more questions there now, Sarah.
One from Horace. Is there any epigenetic influence, i.e. conceived in drought years versus wetter years, or born to years that were in drought affected? Another really good question. And I guess it's, if they're something that I probably can't answer about how it would affect this particular immune trait, but definitely nutrition has a big influence on immunity. If they are on good nutrition, then they've got better able to mount a more effective immune response and can actually tolerate much higher worm infections if they are on good nutrition. So definitely I think there would be potential influences there, but it's nothing that I can really answer on. The only thing I can say about droughts is a lot of this testing was done in the west of Victoria and South Australia during the drought a few years ago. And so that was a good test to show that you can actually, even in drought years, you can still measure an antibody response and get good variation throughout the flock, which is what you want to see. So that was really good. And Mary's question, do you expect variation in antibody responses with different breeds of sheep? Yes, so it's well known that there are breed variations, and with certain breeds being more able to tolerate immune response. So there's definitely breed differences. We have seen in some of the sheep that we have tested that are of the Dorper breed, and they have been bred for resistance based on worm egg counts, that they do seem to have much higher antibody responses than, say, merinos. That's sort of a clear difference when we do that in the lab. There's lots of published research saying that there is breed differences, and particularly breeds that are more resistant to worms, and we do see that with our test as well. Red's question. Another really good question. A lot of we have done sort of measured with farms that have mixed infections, so they'll have Barber's pole and they'll also have trachs and or scale worms, and also the brown stomach worms. And we haven't really seen that there's a higher antibody response because there's a species that's more pathogenic. So it doesn't necessarily, it's not about the pathogenicity of a Barber's pole, like the Barber's pole worm. It seems to all give sort of a similar range of responses.
There's one back on, which I don't think you'll know the answer to, but you may. Why is the Barbavax not used in New Zealand?
I'm not really sure, to be honest. I know that because it is made from adult worms, that there is, potentially there might be some issues with importing it because it's not, recombinantly made, and that's always an issue, perhaps with vaccines. So maybe that's why. I don't really know. I
guess it'd have to be produced here rather than mincing up worms and then shifting it over here would.
I think that might be a biosecurity issue, perhaps.
Couple more in the Q&A.
Have you noticed different levels in flocks that have been selected for WEC? So first, those that haven't. So we haven't looked into the data that much yet, but that's definitely something that we'd like to do, and we'll do that more with once we get a bigger database. But you would hope to see that those flocks that have been selecting for worm egg counts for a long time versus those that haven't done any selection at all, that they would have high levels. The only thing that we've seen and can say with a lot of confidence is just that one flock that has been, and the data I showed that has been selecting for worm resistance based on WEC for a long time, that those ones that were really resistant had higher levels compared to their more susceptible sheep. So that's all we've really been able to confidently see.
Yeah, your original bar graph, we would expect answers that question.
Yeah. That's a really good question. So, Carla's not, and I think Richard's on the line, so he might be able to also answer this, but it's not available in Australia.
For those listening online later on, that question was, is Carla available in Australia? And the answer is no. Oh, I don't think. There may be someone feverishly typing, I'm not sure, but we're getting towards the end of question time. In terms of wanting to find out more, Sarah, what's the easiest way to get in touch with you?
You can just email us at our email that's info@swabtec.com. We can get in touch and talk more about the test results and where we're going in the future. So I've mentioned that we are doing some more on-farm testing just to build up a really good database of tests. So far we've been able to get 4,000 tests with matched worm egg counts across merinos, but also composite breeds as well. So we're just looking to build that up so that we understand the relationship with worm egg counts and other key production traits like DAG and weight, and understand that connection a lot more with this antibody trait. And that's work ongoing with the team at Agbury. And we also, in a few weeks, will look at doing a study where we're going to be following some sheep that we've tested using our saliva test into an abattoir and collecting some of those abomasums and small intestines to actually do a worm count, and also do some mucus scrapings to confirm the correlation between the saliva antibodies and what's happening in the gut. So that should be quite good. We do have to try and dissect the stomachs out at chain speed though, so that will be quite fun, I think. But hopefully we'll have some good data from that as well.
Cool. If there's no other questions, just here we go. Yep. So Richard's just added that currently no one's providing the Carla test in Australia. Fred wants to know, what are the highest results you have seen so far, and from what breed?
So the highest ones we've seen so far is from the Dorper cross breed that has been bred for a long time for worm egg counts. So a lot of those results are on that sort of histogram graph. They're more towards over 20 units per gram, not per gram, units per mil to 30, so the other end of that graph. But I guess, we haven't really we don't have enough data around all the breeds to really have that with a lot of confidence, but that's something that does sort of stand out so far.
I think Freddie's underlying comment is that New Zealand Romneys will be better again.
Thanks, Mary. We're pretty excited to be able to do that. It is quite an expensive study to set up a trial for the amount of sheep that we want to analyse to get good statistical results. So we're excited to be able to do that with an abattoir And this is an abattoir that does provide individual feedback on each sheep. So we'll get quite a lot of data, other data, so that's quite exciting, I think.
Cool. Glad that Freddie has confirmed that he wants Romneys to win. I guess in the answer to that breed question, we will be... Shield has, I guess, shedding breeds, fine wall, and Romneys and the whole shooting match is going to be part of Shield, so there'll be... so we'll have good answers to that breed question. There's a question there from Mary again, Sarah.
So we are measuring measuring the response against something that's secreted by the LEL3 and L4 stage to try and shut down the immune response. So that's what we're targeting with our antibody response test.
So it's both L. Yes, the question is it measuring both L3s and L4s? And the answer is yep. And I learned something on the podcast the other day with you that when they change jackets, they become undetectable.
Yeah, that's one of their good immune evasion skills. Yeah.
To get the full details on that, they'll have to listen to the Head Shepherd podcast. Shameless plug. Cool. Well, we might, I don't know. There's a comment there from Richard. We find shedding breeds have high colour responses, but we have not done any work with mixed breeds in the same environment.
Oh, that's good to know. Yeah, so similar to what we're seeing too, so that's great. Good reassurance. Thanks, Richard.
Sorry, Sarah.
Oh, I was just going to say, yeah, that's sort of one of the, I guess in comparing this shedding breed versus the Merinos, they were in the same area, but they're not on the same property. So that's, I guess, what you'd really want to be able to do is have, and that's what some of the scientific papers have done to show the differences that they have brought in different breeds and raised them under the same conditions to show that. So,
righto, we'll wrap it up there. Having tried a couple of times, but I know we've got another question. So. The one there from Horace. Sheep on organic farms versus conventional, any difference? No drench ever versus some drench?
We haven't tested that at all. Yeah. I assume that they would have to be pretty resistant if they're not to worms if they're not being drenched at all. And I guess that's what a lot of native breeds globally, they tend to not have been exposed to many drenches, and so they've had to build up natural resistance and essentially sort of survival of the fittest to overcome worms. So, yeah, potentially that would be what's happening there. But, again, you'd have to set up a trial and test that.
But you have demonstrated that drenching doesn't impact the antibody response.
Yeah, exactly, yeah.
Just a comment from Sophie, thanking you, and one from me live, thanking you. We will wrap it up there. Thanks very much, all of you that have listened in. The recording of this will be on the hub. Sophie will correct me when, but probably early next week, or knowing Sophie, she'll do it now or something crazy. But anyway, by the middle of next week, I'm sure it'll be there. So if anyone that hasn't been listening that you think should, just direct them towards the hub. They'll jump on there and can watch it from there. It's up tomorrow morning, she's pointing out. So there we go. But yeah, thanks Sarah, and thanks to the team at SwabTec. It's been really, really good fun collaborating with you guys, and looking forward to more of it. And looking forward to seeing what SwabTec can do for the industry and help us as one of the tools to help in the fight on worm resistance.
Yeah, thanks. And thanks again for having me on, and thanks for everyone for coming and asking really great questions.
Excellent. Cheers, righto. We'll leave it there. Thanks all. Good evening.